Answering MCAT questions in under 60 seconds. A research team investigates a point mutation in ATP synthase that reduces its efficiency. They measure the rotational activity of ATP synthase in isolated mitochondria and find a slower rate in mutated cells compared to wild type. Which of the following conclusions is most strongly supported by these findings? A says, the mutation impairs proton flow through the F0 subunit, limiting mechanical rotation. Okay, so what we need to remember is that ATP synthase, or complex 5, is this like large, complex, multi-protein structure that, as part of its structure, has a subunit, the F0 subunit, that allows protons to flow through it as a channel, right? And it's able to then harness the kinetic energy of that proton motion and convert it essentially to rotational energy, okay? So to mechanical energy, which is then used to help drive conformational changes, okay, that help synthesize ATP from ADP to phosphate, which occurs actually in another subunit of the structure, okay? And so if you had a mutation that's impairing proton flow, right, you should have less rotation, and that's what they were observing, that you had reduced or slower rotational activity. And that would definitely occur if you limited proton flow through the F0 subunit, right? Because that's actually what is driving that rotation, right? So A sounds like the correct answer, but we'll check the other ones as well. So B says, the mutation increases proton leakage across the membrane, enhancing ATP turnover, right? Well, that is kind of describing maybe something that's allowing protons to move freely independent of ATP synthase, but you wouldn't observe slowed rotational activity per se. Okay, you would observe overall less ATP synthesis per proton units, right? But the actual rate of rotation shouldn't really change, okay, when you actually have protons moving through the channel. Now C, the mutation enhances binding of ADP and phosphate, leading to increased rotational speed. Well, that's just wrong because they actually observe less rotation, okay, so that doesn't make sense. And then D, the mutation disrupts electron flow in complex 4, indirectly affecting ATP synthase. Well, again, if you have a mutation in complex 4, that shouldn't change the ATP synthase itself, right? It'll change how many protons you can get into the into the intermembrane space, but they're specifically finding in this experiment some sort of intrinsic problem with complex 5 or ATP synthase. So that's not right. So A here is the best answer.